Good afternoon, everyone. Thank you for your invitation, and I have extensively collaborated with Cellecta in the last 4 years during my postdoc study. So throughout my Ph.D. and the postdoc study, I have been interested in characterizing the genomic drivers that drive breast cancer progression and therapy response. So thank you everyone for giving me the opportunity to share my work with you today. So very brief introduction of breast cancer is the most popular cancer in women in most of the areas as shown in pink, and it's the second leading cause of cancer deaths worldwide. And for breast cancer, it can be divided into several groups based on the expression of estrogen receptor, progesterone receptor, and also HER2, whether they have amplified the HER2. So for luminal cancers, they have the marker of estrogen receptor. For HER2, they have amplified the HER2, and the basal-like cancer cells, they are negative for PR, ER, and also HER2. So today my talk will only focus on the luminal cancers, which they have the marker of estrogen receptor. So for ER-positive luminal-like cancer, they originate from the luminal layer of the normal mammary gland, which requires the ER transcription program. So whereas the basal-like cancer that acquire the basal-specific drivers, then it causes the generation of basal-like cancer. So briefly, ER binds to its ligand estrogen together with other pioneer transcription factors like FOXC1, GATA3, which are very essential for the fate of luminal cancer. So if they have uncontrolled ER transcription program, it will generate a luminal-like cancer, which are ER positive. For ER-positive breast cancer, they are primarily treated with endocrine therapy, including tamoxifen, which are ER modulator, or aromatase inhibitors that block the production of estrogen, which is the ligand of estrogen receptor. Or you can use the SERD, which is selective estrogen receptor degrader, for example, fulvestrant. So in this study, I only focused on fulvestrant, which is SERD, and very much popular used in the clinic. However, there's a painful truth that is the drug resistance in ER-positive breast cancer, and since there are about 70% of breast cancer are ER-positive, so it's a very, very important research priority for understanding the mechanisms of resistance to endocrine therapy. So to understand drug resistance as well as other clinical relevant cancer problems, our institute launched a large-scale clinical sequencing initiative named the MSK Impact. It is a targeted exome sequencing which only targets about 500 actionable genes. It's not the whole genome, it's just target about 500 actionable cancer genes. In contrast to TCG, which only focused on the primary and the pre-treated tumors, MSK Impact also sequenced the heavily treated advanced and metastatic cancers. Up to date, there are about 40,000 cancer patients which had been sequenced at Sloan Kettering, and most of the data is available to the clinic. So you can just go to the CBI portal to check all the data. So in our lab, we only focus on breast cancer. So we compared about 500 endocrine therapy resistant tumors versus 500 treatment-naive tumors. And the resistant tumors could be divided into 4 groups, so which allowed us to really stratify those tumors. So for example, the tumors with the ESR1 mutation, so ESR1 is a coding gene of ER, and this has all been shown to cause resistance to endocrine therapy in the clinic. So we found about 18% of the patients with this mutation. And we also found the tumors with alteration of the MAP pathway, and also about 9% of the patients they show alterations of the transcription regulators. That means a kind of epigenetic regulated mechanisms of resistance. And of course, we also have a lot unknown mechanism of resistance. So in this paper, we only focus on the MAP pathway, but for my project, I aimed to determine to determine epigenetic-based mechanisms of resistance to endocrine therapy. To achieve this goal, I employed the functional genomics approaches to really do this. Because I had a lot of, some experience with to identify resistant mechanisms of, for example, triple-negative breast cancer to PARP inhibitors during my Ph.D. So basically, we just employ functional SHR screens and then identify genes that may mediate drug resistance. PARP inhibitor resistance of BRCA1 deficient tumor cells. When I joined the lab at Memorial Sloan Kettering, and CRISPR was very hot. That's why we turned to CRISPR screens to really do this project. So first we asked Cellecta. We collaborated with Cellecta to generate this human epigenome CRISPR knockout guidance library, which targets about 914 genes. And each gene we have about 12 guidance, because at that moment I didn't know how many guidance we need to really have efficient, to really because we do not want to miss any genes of interest. Of course, you also need some control guidelines, including the essential gene controls and also negative gene, and also some non-targeting controls to really perform these screens. And in total, we got this 11K, so 11,000 guidelines library. And if you want to, I'm happy to share these things. Okay, so briefly for this, because Paul just talked a lot about the strategy of these screens. So briefly, we package the guideline library into lentivirus and then infect the Cas9 transducing cells at a very low MOI to really allow us to only get one guide RNA per gene, because per cell. We do not want a lot of guide RNAs per cell. And then the positive selected cells of the pure medicine were expanded for about 2 weeks to really allow efficient genome editing. And then the cells with gene knockout, these cells were subject to either DMS treatment or fulvestrant treatment. And of course, you need to isolate the genome-DNA from each group to really amplify the barcode by PCR to retrieve the guidance sequences. And through some bioinformatics analysis, you could easily get which guidance are enriched and which guidance are selectively dropped out. Yes. So, and here is the data of the screen. I think, if you see this figure, I don't think we need any criteria to select the top hits, because apparently this is the very, very top hits. So from this string, we just plotted the log2 mass of reads in the DMS group against the log2 abundance between the fulvestrant group and versus DMS group here. And we could clearly see that the ARID1A guidelines, ARID1 guidelines were selectively enriched in the fulvestrant treated group versus the DMS group, suggesting that ARID1A loss may cause the resistance to endocrine therapy. Here is the fulvestrant resistance in this cell line. And very important is we almost did not lose any guideline, because normally we do 1,000 times coverage from the beginning to the end, and we could recover at least 99.9% of the library after sequencing. So you may ask, what about the whole genome screen? So in this paper, we only focused on the epigenome screen, but of course we also did the whole genome screen, and we can clearly see that ARID1A guidelines were also enriched in this whole genome screen, and we could see that we saw six out of 8 ARID1 guidelines were selectively enriched in fulvestrant treatment setting, and here are the criteria for hit selection, and we could see, so at least 5 out of 8 guidelines targeting the same gene were enriched, and they belong to the top 2K guidelines out of 150K. So it's only top 1.3. So is a very, very stringent criteria to select the hits. But since we already used the epigenome screen for this paper, so we can use this data for another paper, another project. Yes. Okay. So what's ARID1A? So ARID1A is the component of the human SWI complex, and which are very important to regulate chromatin accessibility and gene transcription by regulating the chromatin structure. So it's a chromatin remodeler, and this chromatin complexes can further be divided into two subfamilies. One is buff and then one is p-buff. Each of them contains about 15 members. Yes, so we just need to remember that ARID1A SWI is the chromatin remodeler that regulates the chromatin structure and the chromatin accessibility. Okay, so because we are a cancer lab and we are at Memorial, so we always check whether ARID1A is clinically relevant. Importantly, importantly, we observed that ARID1A is the most popular alterated gene. According to MSK impact, so we can say about 6% of ARID1A show alterations in breast cancer, and from this we can clearly see that most of the alterations are truncations or inactive mutations, which suggests that they are kind of lose the protein expression or protein function. So it's kind of a tumor suppressor. Yes. To validate the results in vitro, I knock out ARID1A by 4 distinct guidelines. We always order these guidelines from Cellecta. So I knock out this ARID1A by 4 distinct guidelines, and we could clearly see the knockout of ARID1A. By crystal violet experiment, we can clearly see that ARID1A lost cells. They kind of cause resistance to endocrine therapy fulvestrant compared to the ARID1A wild-type cells. To validate the results in vivo, I oscopically transplanted the ARID1A wild-type cells and ARID1A knockout cells into ASCB mice, and then I treated the tumors when the tumor reached a volume of 50 cubic millimeters with fulvestrant. So from this tumor growth curve, we can clearly see that ARID1A knockout cells, they show reduced sensitivity to fulvestrant compared to the ARID1A wild-type cells, wild-type tumors. Again, we are at Memorial Sloan Kettering, so we like to find the clinically relevant problems. That's why we ask the clinicians for help to really analyze our MSK cohort, the ER-positive breast cancer patients who were treated with endocrine therapy. Here we treated with SERD fulvestrant, and from this Kaplan-Meier curve, we can clearly see that for the ARID1A for the ER-positive breast cancer patients with ARID1A alterations, they have much shorter PFS, so much shorter progression-free survival compared to the ARID1A wild-type patients. This is a black and white experiment. So far we have already determined that ARID1A causes resistance in vitro, in vivo, and also in the clinic. So next we want to know what about the mechanisms of resistance caused by ARID1A loss in ER-positive breast cancer. Yes. So to address this question, we first just harvest the ARID1A knockout cells and ARID1A control cells for RNA sequencing. So at the beginning, we have no idea about the mechanisms. So we first did the RNA-seq, and we were very excited to see that the basal through gene set enrichment analysis, we were very excited to see that the basal markers were very much enriched versus the luminal marker, suggesting that luminal to basal transition. So from the luminal signature to the basal signature upon ARID1A knockout. And this is very important in the clinic because according to our clinician and also as well as other clinicians, they could say about 18 to 25% of ER-positive tumors become ER-independent. Become basal-like cells and endocrine therapy resistant by switching to a basal-like signature. of course, the mechanisms behind are really largely unknown. So we were excited that to see that maybe we found a normal mechanism of luminal to basal transition by ARID1A loss. So from ARID1A loss, you see the switching from luminal to basal. That's why for the luminal specific therapies, they do not work anymore. Yes. So next the next question is how does ARID1A loss mediate the reprogramming of a luminal to basal fidelity? The transition. So as I showed you just now, ARID1A is an important component of the SWI state complex, which is chromatin remodeler. So the first question we want to address is whether ARID1A loss really affects the chromatin binding of the SWI state complex. To address this question, we performed the ARID1A chipstick, as well as two other subunits of the complex, the BRG1 and BAF155, by chipstick in ARID1A knockout cells and ARID1A wild type cells. So here is a very good control that you see in the ARID1A knockout cells, you see marked decrease of ARID1A binding to the chromatin, suggesting they are really ARID1A binding sites because ARID1A is knocked out. So you must see ARID1A decrease the ARID1A binding to the chromatin. If you cannot see it, that means those binding region is not correct. And importantly, furthermore, we could see marked decrease of BAF155 and the BRG1 binding to the chromatin, suggesting that ARID1A is very important to bring the SWI stem complex to the chromatin. If ARID1A is lost, the whole complex cannot go there anymore. Not only ARID1A, but the whole complex. So now we confirmed that the SWI we observed that the SWI stem complex cannot go to the cannot go to the chromatin anymore, and we knew that the SWI stem complex is very important for regulating the chromatin accessibility. That's why the next question is whether the impaired SWI stem chromatin binding affect chromatin accessibility. So. To address this, we performed a taxing, which could examine the chromatin accessibility globally. So consistent with the role of ARID1A and SWI stem in regulating chromatin accessibility, we could see marked decrease of chromatin accessibility in ARID1A knockout cells compared to the ARID1A wild type cells. Importantly, when we integrate the ataxic data with the chipstick data, we could see the co-occupancy of the ataxic data and also the chipstick data, suggesting that this impaired chromatin accessibility is really due to loss of SWI stem binding to the chromatin. So because they are the same chromatin region, we just integrate the ataxic data and the chipstick data. Yes. Okay. So far now we found that chromatin accessibility is lost in ARID1A knockout cells, and now we need to think about some functional studies. For example, as we all know that the chromatin accessibility is really closely tied to transcription factor binding and gene transcription. So if the chromatin is shut down, it's not open, transcription factors cannot go there and cannot be functional to really regulate gene transcription. That's why the first thing is what we want to know. The next thing is what we want to know which transcription factors are affected in those impaired chromatin binding, impaired chromatin accessible sites. So it's very easy. We just need to perform, of course, the bioinformatician, we need to perform multiple analysis. And surprisingly, we found that the FOXC1, GATA3, and the ESR1, ESR1 is the coding gene of ER, they are strongly enriched in those impaired regions, suggests that they cannot go there anymore because the chromatin openness is impaired. And now you might still remember that the ER and the FOXC1 GATA3 are very important for the fit of luminal cell identity. So if they are not functional, you cannot maintain the luminal cell anymore. They will probably switch to basal-like cell. And so that's why that's why the next thing is we need to check whether the FOXC1 and the ER really is cannot go to the chromatin because the chromatin shut down. So to address this, we perform the chip-seq. So again, FOXC1 chip-seq and ER chip-seq to see whether they can go to that region anymore. And we could see it's very clear that FOXC1 and ER they cannot go to that region anymore. So because that region is closed. So these transcription factors cannot go there anymore in ARID1A knockout cells compared to the ARID1A wild-type cells. So the last thing is the last and also the most important thing is whether ARID1A loss change ER-dependent transcription. Because all those above experiments, we only have a prediction that they change the luminal identity, but we still have no proof yet. If we want to say, if we really want to confirm that ARID1A knockout switch the cells from luminal to basal, we need to confirm that the cells are not ER dependent anymore. So to address this, we just simply did the RNA-seq experiment. So first, we deprived the estrogen from the ARID1A wild-type cells and the ARID1A knockout cells. So we just deprive the estrogen; they have no estrogen, and then we stimulate the cells with estrogen and of course also DMSO. So we need to stimulate the cells with estrogen to really stimulate the ER transcription. And so for the ARID1A wild-type cells, with the estrogen, you can see the transcription change in ARID1A wild-type cells. However, in the ARID1A knockout cells, the transcription the ER-dependent transcription is totally messed up. So they cannot depend on the estrogen receptor anymore. Yes, so it's globally, and we could say about 1,200 genes which were significantly changed in ARID1A knockout cells upon estrogen stimulation, suggesting that ARID1A loss really reprograms the ER-dependent transcription. So you cannot have a classical ER transcription in ARID1A knockout cells anymore. Yes. So to conclude, as I showed you just now, so ER-positive breast cancer, luminal breast cancer, they evolve from the inner side, I mean the luminal layer of the luminal gland, and if you have uncontrolled ER transcription program, you have ER-positive breast cancer, which are luminal specific. That means you have ER-positive things. And then for this ER-positive, you can use endocrine therapy, which target the ER to really kill the cells. However, in the clinic, we frequently observed that ER-positive cancer cells can be killed, the light population can be killed by endocrine therapy. However, there's still a small fraction of ER-positive breast cancer which could escape from endocrine therapy and acquire resistance, but we didn't know the resistance mechanisms to endocrine therapy. The resistance mechanism is largely unknown. So in this, in our finding, we observed that ARID1A loss, ARID1A knockout cells, they could switch the cell identity from luminal to basal, which could escape from the endocrine therapy, ER-positive specific drug treatment, for example, endocrine therapy. Yes, so to conclude, for our acknowledgement, I would like to thank my boss Jose and Mauricio from MSK, and we have a very nice collaborators in that, and people from Sloan Kettering who are expert in bioinformatics analysis, and also people from Dr. Faber. Yes, and I also would like to thank my funding from Stanford to Cancer, and also this project was also supported by a number of other funding resources. Thank you for your attention, and I'm happy to take any questions.